We're going to get started. So it's a pleasure to introduce Carol Sortwell from our friendly neighbor at Michigan State. Carol's in the Translational Neuroscience group, actually the Grand Rapids campus from MSU. Carol's a professor of translational neuroscience and is going to talk today about something which she has done really excellent work on, which is the development and use of models of Parkinsonism, both in the sense of models that are useful for investigating circuit level changes, but also epigenetic mechanisms. This has, I think, historically been not only important, but in a sense somewhat neglected, as people tended to use models in a fairly cavalier manner. And Carol has really done first-rate work in terms of characterizing and developing models in a really rigorous conceptual way. So thank you.
Thank you. Thank you very much. I'm glad that I made it back here. I was supposed to come right before the election and was ill. So thank you for having me back. I was thinking about the drive over here when I was listening to the RFK Senate confirmation hearings that if you had told me when I was supposed to come before, that's what I would be listening to on the way over. When I actually rescheduled, I wouldn't have believed you, but it's nice to be here to talk about science. It's a nice distraction. So before I begin, just if anyone here in the room has a question or on Zoom, you feel free to interrupt me mid-slide so we can, answer it right then and there. So as Roger mentioned, I've spent a lot of time studying animal models of Parkinson's disease, and in particular, this model I'm going to talk about today, the alpha-synuclein preformed fibril model, and I've been fortunate to be given funding just to do that, like to characterize it extensively, both from NINDS and the Michael J. Fox Foundation. So here's an overview of what I'd like to talk to you about today. First, I'd like to just sort of ground ourselves about what we're looking for, about what makes a good animal model of Parkinson's disease, a little bit about how we generate and execute the PFF model, and then the bulk of what I'm going to talk about today is like sort of what we've learned about the consequences of inducing this model almost exclusively in rats, and I work primarily with these with rats. And then last a little snippet about some ongoing work that we have. We're now applying what we've learned in this model to perhaps developing some therapeutics, as well as expanding to models of co-pathology. So before I begin, I'd like to say that, my work is like the bricks, the building blocks of my research has strongly related to wonderful collaborations that I've been able to have. So I wanted to specifically list them here, working with excellent PET imagers, people who are expertise in microdialysis, my former postdoc, Joe Patterson, who is now my colleague, who is an excellent geneticist, as well as, has become the source of creating our own fibril material. And then this work started with Kelvin Luck from Virginia Lee and John Trojanowski's lab, like physically coming to our lab, holding our hands in the surgery rooms and making sure we did things right from the get-go. Excellent behavioralist, Sheila Fleming at Neomed, and then a lot of insight and advice from Malou Tanzi, who's an expert in neuroinflammation, AD and PD. And then, of course, always got to remember the funders. So when we're talking about any animal model, but in particular when we're talking about Parkinson's disease animal models, what makes a good model, basically those criteria fall into three broad categories of face validity, or how well the symptoms and the pathology in the model, completely recapitulates what's seen in the human disease. And then construct validity is what we, at least to the best of our understanding, is believed is causing the disease. Is it similarly what's being used to cause the disease-like state in the animal model? And then predictive validity, which is, how well, if there's therapeutic treatments that work in the disease, either for symptoms or disease modification, how well do those same therapeutics work in the animal model of disease? So for Parkinson's disease, for face validity, we're talking about the two main pathologies of the disease, alpha-synuclein Lewy body-like inclusion formation, as well as, at the very least, nigrostriatal degeneration. And then I would add a third hallmark of Parkinson's disease, as well as other degenerative diseases. We're appreciating more and more the role of neuroinflammation. And then the features of motor dysfunction and cognitive dysfunction, and especially in terms of later stages of Parkinson's disease. And then for construct validity, we're talking about a model of Parkinson's disease in which the alpha-synuclein inclusion formation precedes the subsequent degeneration. And then lastly, for predictive validity, where we don't have disease-modifying therapeutics that we can test, we certainly have many dopaminergic anti-Parkinsonian medications, most primarily levodopa or Sinemet that's used in the clinic. And so do the symptoms in the model that you're creating, can they be reversed by levodopa administration, for example? So these are the questions that we've asked using varying animal models across the years. And I've used, neurotoxicant models, primarily 6-hydroxydopamine. I've done a lot of work with alpha-synuclein overexpression models, both viral vector mediated and some transgenic. And so these are how these models stack up in terms of just using the same metrics about being a good model for Parkinson's disease. So we know that in neurotoxicant models, they're great at inducing nigrostriatal degeneration, and as well as studying the consequence of severe loss of dopamine in the striatum. There's a couple reports that suggest there could be some alpha-synuclein pathology, but that's mostly just in some cases maybe some phosphorylated alpha-synuclein, but never like true Lewy body-like inclusion. There's definitely a lot of neuroinflammation, but that's likely just related to a massive acute destruction of dopamine neurons. And so how analogous that is to the disease state is not quite clear. And there's definitely some examples of motor and cognitive, definitely motor dysfunction and cognitive dysfunction in neurotoxicant models. And then for construct validity, I don't think these neurotoxicant models get a good rating in terms of like alpha-synuclein pathology is not what's driving the degeneration. It's the specific oxidative stress toxin that's used. But certainly you can get levodopa responsive motor symptoms in these neurotoxicant models. Alpha-synuclein overexpression, transgenics, they are great models if you want to study alpha-synuclein inclusion pathology and neuroinflammation, and in some cases, motor and cognitive dysfunction. But very rarely do they show any level of nigrostriatal degeneration. In comparison, alpha-synuclein overexpression models, they have a lot of nigrostriatal degeneration. They have some form of alpha-synuclein inclusion pathology, but again, not true Lewy body-like inclusions. They have a lot of neuroinflammation, and they have cognitive dysfunction and motor dysfunction. Again, in construct validity, both of these models are not ideal in terms of like inclusions driving the degeneration of the neurons that had them. And then in terms of predictive validity, you can definitely reverse some alpha-synuclein overexpression by viral vector. Some of those motor phenotypes can be reversed with levodopa. I've not seen, it's rare that you get that robust motor dysfunction in the transgenic models, and so therefore it can't be reversed. So having shown you that, I guess sort of my pitch that I'd like to make to you today is that I think the alpha-synuclein preformed fibril model, both in mice and rats, comes closest to satisfying the criteria that I just showed in terms of recapitulating all the features. I'm very aware that a model is a model and not the disease, but models can be very useful if they are appropriate in order to get at disease mechanisms that then definitely need to be validated in clinical studies. So this work started out for us after seeing Kelvin Luck's and Virginia Lee's publication in Science, where the first time they showed in non-transgenic mice that if they injected these PFFs into mice, wild type mice, they could induce this inclusion pathology and degeneration. Before that, there had been work in cell culture where this had been shown, that they showed that there had been work in overexpressing, alpha-synuclein overexpressing mice. But this was what really made us sit up and take notice when this was occurring in wild type mice. And so we worked with Kelvin to come into our laboratory and to see if we could extend this model into rats. And then luckily, we were able to, the first time out, get it to work because he was there. Then we were off to the races with all the studies that I'm going to show you today. We love to work with rats over mice for a number of reasons. Their larger brain and body size for a number of practical reasons, sample size, the ability to image, the ability to, for example, I can get CSF from a rat quite easily. It's from a mouse in any large amount. And then they do have more complex motor, and I would add also cognitive behaviors that can be studied, and they do have greater synaptic complexity than mice. So for those reasons, we primarily work in rats. This is how we generate the rat alpha-synuclein preformed fibril model. We generate, grown from bacterial cultures, we generate alpha-synuclein monomer of varying species, mouse, human, rat. That monomer is then fibrilized, adding heat and agitation over time. And those result in the formation of these preformed fibrils. These fibrils are validated by a number of ways. Joe Patterson in particular has a lot of papers that describe this, but in particular, they're, sedimentation assays, electron microscopy, to see that the conformation of fibrils are accurate and consistent. It's very important to sonicate the fibrils prior to use, before you inject them or apply them to culture. They need to be sonicated to a particular size in order to work. This is a step that has been sort of like the problem of many investigators that didn't look at the fine print, and they just think sonication is sonication, inject them into an animal. 6 months later, look, nothing's happened. And it's because the fibrils weren't sonicated to the appropriate size. These injections of fibrils can go to the striatum, the substantia nigra. People have looked in multiple other sites. Primarily, we do our work by injecting in the striatum, but in some cases, we do inject in the substantia nigra. What happens after these fibrils are injected is that they're taken up into neurons, and where inside they undergo incomplete autophagic degradation, and then they corrupt and hyperphosphorylate the endogenous alpha-synuclein. So if a neuron or cell doesn't have endogenous alpha-synuclein, no inclusions will form. After a few days, you can't even detect the initial fibrillar seed material itself. So what you're seeing with the pathology is the formation, triggered the formation of these endogenous alpha-synuclein inclusions. And that's dependent upon the amount of alpha-synuclein in the neuron that took it up, as well as time. And importantly, as indicated by the science paper, this can happen in the context of normal alpha-synuclein levels. And I think it's important that in my opinion, I think some of our understanding about pathogenic mechanisms of Parkinson's disease, some of those mechanisms that have been implicated might be red herrings, because we've been looking at models in which there's massive supraphysiological overexpression levels of alpha-synuclein, which again might not be disease analogous. As proof of principle that you need endogenous alpha-synuclein to cause the inclusions to form, if you do these same experiments in knockout cells or knockout animals that don't have alpha-synuclein, you will get no toxicity and you will get no alpha-synuclein inclusion pathology. I mentioned the considerations about size. The fibrils need to be below a certain length in order to be physically be able to take it up by the neurons, and people have determined that that's about 50 nanometers. There's different efficiency in terms of how well, especially in rodents, different fibril species will seed. So mouse fibrils will seed very well in mice and rats, but human fibrils, they'll definitely still seed, but in a wild type animal, you'll get a much lower burden of inclusion pathology and degeneration. And then often we talk about control conditions. people either use an equal volume of vehicle, and in some case, an equal quantity of alpha-synuclein monomer as a control condition. So you can control for surgical artifact. In the case of monomer, control for the actual protein itself. So these are important quality control measures that we go through to determine whether the inclusions that are being formed that we're seeing in the model are sharing appropriate features with Lewy bodies. And so what you see in rats and mice is that after PFF injection, you get phosphorylated alpha-synuclein, you get oligomeric forms. The inclusions are oligomeric and they're fibrillar, and they're thioflavin positive. They co-localize with ubiquitin and P62. And importantly, they're proteinase K resistant. And so here's some examples of PSIN serine 129 staining in nodule neurons 2 months after PFF injection, thioflavin positive, and are resist proteinase K digestion. In some work conducted in culture in Hilal Lashul's lab, this is an excellent paper from 2020. They actually looked at the properties and the time course of inclusion formation, and looking from, using RNA-seq, using proteomics, and just trying to understand what happens once you are PSIN positive and you are determined to be an inclusion. Are there later conformation states that are happening, and how does that relate to Lewy body-like formation? And so basically, with the addition of PFFs to asyn monomer, the first stage is phosphorylation and oligomer formation, and then fibrillar formation, and then aggregate formation. And up to this point, essentially, the aggregates are almost 100% phosphorylated alpha-synuclein. But over time, with maturation, these inclusions, portion of them begin to be more Lewy body-like, in which they've developed this property of their sequestering organelles and lipids into the alpha-synuclein aggregates. And that's an important characteristic that's shared with Lewy bodies. And also around this time, we're able to pinpoint this time and beyond that's when neurotoxicity starts, cell death pathways in the neurons start to be activated. So it's not just that aggregates are formed that are toxic, but there's the process of maturation of these inclusions to be more Lewy body-like is associated with cell death. So practically, when we inject these fibrils into the striatum of rats, we see widespread accumulation of alpha-synuclein pathology, and basically dictated by the injection site and what's innervating the injection site. So if we inject into the striatum, there's bilateral innervation from the cortex to the striatum, and so we can see inclusion pathology in both sides of the cortex. There's unilateral innervation of the amygdala and of the substantia nigra. So basically you see ipsilateral inclusion formation in those structures. We see that the inclusions peak, and in the case of the nigrostriatal neurons, you can see initially first see a loss of TH phenotype or DAT phenotype in those neurons. And some important work by Vivek Uni's laboratory showing that if you watch at least cortical neurons with these inclusions over time, longitudinally with two-photon microscopy, you can see that it's the inclusion-bearing neurons themselves that are the ones that are degenerating, whereas the neighboring neurons that don't have inclusions survive. So it's a direct link between, it's not an issue of clearing alpha-synuclein of those neurons. It's a direct association between inclusion formation and ultimate degeneration. This is what we see 1 to 2 months after preformed fibril injection into the striatum. This is a PSIN serine 129 stain, where you can, it's a marker where you can definitely appreciate the topography of the substantia nigra. In the cortex, also at 2 months, there's abundant inclusion formation all over the cortex. When we look at the time course of inclusion formation, We see a peak that's between 1 and 2 months. And then over time is when we see the number of inclusions in the NIGRA decrease. We don't have such a detailed time course in the cortex, but we know that over time, again, you lose peace and inclusions in the cortex. And this is important because inclusions are decreasing over time because the neurons that were initially seeded are degenerating. You can see co-localization of PSIN with TH-positive neurons. And so this is sort of the early profile of peak aggregation pathology after intrastriatal injection. If you look across many structures over 2, 4, or 6 months after intrastriatal PFF injection, I guess I'll focus you here on, again, here's the NIGRA at 2 months and then declining over time as these neurons die. Here's the cortex peaking early and maintaining inclusion pathology for a longer period of time. So it's somewhat indicative of resilience of cortical neurons to inclusion formation relative to a substantia nigra neuron. And then another interesting profile is what we see in the striatum. Even though we're directly injecting into the striatum, inclusions in the medium spiny neurons themselves in the striatum do not peak until later time points. And so this is an indication, you can see this again, 2 months, 4 months, 6 months. We know from cell culture studies that the fibrils are being taken up at the same time at the same rate, but we also know that the striatal neurons have less alpha-synuclein compared to other cell types, certainly compared to nigral dopamine neurons and cortical striatal neurons. And so this appears to be dictating how quickly inclusions are formed. One thing I'd like to specifically talk about is that some people think of this model as a model of second order prion-like spread. And I'm here to say that is not what we have seen. What we see is that, neurons where you inject the fibrils take up the inclusions or take up the fibrils and form inclusions. Neurons that innervate where you've injected take up the inclusions. So again, we're talking about cortical striatal neurons, nigrostriatal neurons, and amygdalostriatal neurons. We don't see, and we've looked up to 14 months past PFF injection, evidence that structures that weren't initially innervating the injection site are forming alpha-synuclein inclusions. So we don't have any evidence that there's release of pathological asyn in a prion-like fashion that's causing second order transmission. So for us, this is a model of initial seeding of inclusions and what are the consequences of those. So because the area where you inject and the innervation to that area will dictate where you see the alpha-synuclein pathology, you can do things like compare a nigral injection of PFFs and where you'll see the substantia nigra, you'll see the amygdala, but because there's very little innervation from the cortex to the substantia nigra, you do not see any cortical pathology in contrast to the cortical pathology that you see with striatal injection. And so you can study questions, and one thing that we're working on right now is under conditions of relatively equivalent nigrostriatal degeneration and inclusion pathology, what is the influence of cortical pathology on behavioral measures and other measures? So that's sort of like the baseline characterization of what's happening when in terms of inclusion pathology in this model. And then so the remainder of my talk, I'd like to talk about, what are the consequences of this inclusion information? So we've looked at the impact on the nigrostriatal dopamine neurons, on microglia and astrocytes that are immediately neighboring the inclusion-bearing neurons. Some studies with collaborators on dopaminergic transmission. We've looked at the impact on gene expression, and then we've done some studies with Sheila Fleming on motor and cognitive. This is the time course of progressive nigrostriatal degeneration following the PFF injection in the striatum. And the basic pattern that we see is progressive loss of TH over the course of 2 to 4 to 6 months. So the number of nigrostriatal dopamine neurons counted by stereology. What's interesting is that you can't just count for TH, because in this model, there's an early phenotypic loss that's revealed if you actually just count with an overt neuronal marker like QC or Pressel violet or NUN, that you can see that even though these neurons lose TH early, they don't actually begin to undergo overt degeneration until later on. So the early phenotypic loss followed by degeneration. So this is just comparing 6 months in a control and 6 months after the injection. You see a similar pattern somewhat in terms of innervation of the striatum. And so you can see, again, a progressive loss of TH over time, progressive loss of dopamine transporter. And that's here showing 6 months after unilateral injection, looking at dopamine transporter immunohistochemistry. With Sheila Fleming, we've looked at a number of behaviors. And so this is where we get to what are the limitations of this model. And that as you'll notice, we're kind of tapped out here at about 50% block of nigrostriatal dopamine neurons. And therefore, the behavioral, dysfunction that we see in motor function is relatively subtle compared to like a 6-hydroxydopamine lesion or other lesions where there's almost complete unilateral denervation, complete loss of dopamine in one hemisphere of the striatum. But we can pick up certain subtle phenotypes. We can see change in these changes that are associated with akinesia, in movement initiation, significant deficits with PFF injection, and that tested also in the adhesive test contact, where you can put the stickers on the rat's paws and take, it's a sensory motor test where they have to recognize they have the sticker. How long does it take them to remove the sticker? There's deficits there. We see deficits in some aspects of the cylinder activity, in the cylinder task. We are starting to look more at cognitive deficits, and I have a little bit more data about that in a later slide. We can see some deficits in object recognition, investigation time. And then importantly, in terms of one of the criteria for a model, in the behaviors in which we can see a motor deficit, we can reverse those deficits with levodopa. And so, and this is under conditions of this about 60 to 70% loss of striatal dopamine. This is work that I was able to conduct with Vesna Sassi at the University of British Columbia, where we did a longitudinal study looking at 2, 4, and 6 months after intrastriatal PFF injection to look at changes in fluorodopa signal in the striatum, as well as changes in DAT expression using 11-CMP. And the story that we found is that there are deficits ultimately with fluorodopa, so deficits in dopamine storage and synthesis, but these are relatively minor compared to this progressive loss in dopamine transporter that we can see early and becomes of greater magnitude. And so this was, Vesna does a lot of work with PET imaging in Parkinson's disease subjects and early Parkinson's disease subjects. And these results of this relative preservation in dopamine synthesis and storage, but earlier deficits in DAT mirrors what's seen in early Parkinson's disease subjects. So, I think we can think of the PFF model, even in its optimized condition, is a model of, I think of it as a model of earlier stages of Parkinson's disease. And then this is just showing loss of DAT over time in PFF injected rats compared to controls. Working with Chris Bishop, who's an expert in microdialysis, we asked questions about, when is dopamine release being impacted in the striatum in this model, and looking at behavior, forelimb impairment, basal dopamine release, and potassium evoked dopamine release. We can see at 4 months that in, where you can't see forelimb deficits, you don't see changes in basal dopamine release, and you're beginning to appreciate loss of evoked dopamine release. These are all, these no basal loss and barely any evoked dopamine loss. It's sort of matching the fact that we can't pick this up any sort of behavioral deficit. But if you look at 8 months, then you're seeing behavioral deficits, you're seeing loss of basal dopamine, and you're also seeing loss of evoked dopamine. So this is just looking at, this is a normal spike after potassium in the striatum to see how much dopamine is increased in the striatum in a control animal. And this is how much it spikes in a PFF animal after 4 months. So not significant yet, but you can, there's a hint of a deficit. Had we been powered greater, perhaps it would have been significant, but definitely by 8 months, there's loss of the spike in dopamine release. So a lot of my work in recent years has been looking at the impact of alpha-synuclein inclusions on neuroinflammation. And so, for us, we're very interested in trying to disentangle what's the neuroinflammatory signal associated with the formation of a Lewy body, or in this case, a Lewy body-like inclusion. How is that a separate, or is it a unique signal from just loss of a neuron, which we know any sort of neurodegeneration is going to cause neuroinflammation. And we sort of like to talk about concepts of like, can we put the suspect, the neuroinflammation suspect, in the room before the actual murder, before the degeneration? And so this model, because of these unique stages and protracted stages of formation of inclusions, you have some time, and then there's degeneration, allows us to sort of look at these different stages and ask different questions. So mostly we've been looking at questions of when and what type of microglial activation is there relative to what's happening in the pathology of this model. And we've done some work looking at astrocytic activation. So we know some basic things about the model. We know that at 2 months, when we're at peak inclusion formation in the substantia nigra, we see more microglia, we see larger microglia, we see increased expression of MHC2, indicating antigen presentation, certainly indicating activation. This is just comparing PBS injected, this rat was injected with PBS in the striatum. There's normally very, very little MHC2 expression in the substantia nigra compared to a rat that received preformed verbal injection in the striatum. We also like the concept of the fact that we're doing an injection, we have injection artifact in the striatum, but we're looking at neuroinflammation in the nigra. So at least we're getting rid of the compound of the surgical artifact of the injection, which is very important when you're looking at neuroinflammation. This is just showing the pattern of the more inclusions you have, the more MHC2 expression you have. So here's 2 months, 4 months, 6 months inclusions, and you can see a decrease in MHC2 expression over time. We can see actually that the MHC2 positive microglia are interacting or appear to be interacting with inclusion bearing nigral neurons. And when we actually quantify activated microglia with MHC2 over time, you can see a specific spike in MHC2 during the peak inclusion formation time. And surprisingly, during the period of degeneration, when we can actually capture loss of dopamine neurons, this MHC2 expression is relatively low. So this tells us that the signal is associated with the inclusions themselves and not the degeneration, or not as much to the degeneration that's occurring. This is also, just disease analogous in terms of like in the Parkinsonian brain, adjacent to Lewy bodies, there are MHC2 immunoactive microglia. And this also similarly, like we see, correlates with the burden of alpha-synuclein. Looking at astrocytes, we see a similar impact in that at the time of peak inclusion pathology, we see increased expression of GFAP, we see increased branching and length of the astrocytic processes. And again, this GFP expression positively correlates with the amount of PSIN burden. So we have this basic phenotype of activated microglia, activated astrocytes, but we wanted to understand functionally what's the phenotype, what are the transcripts that are, what are the genes that are associated with these inclusions, with these microglia that are in the immediate adjacent vicinity of the inclusions. And so we've done a lot of work using droplet digital PCR to quantify gene expression levels and localization of the actual upregulated mRNA to microglia or astrocytes to confirm the gene expression changes that we've been seeing. And we can identify a suite of genes that are upregulated in the inclusion-bearing substantia nigra at 2 months. Most pronounced genes that we see are CD74, which is a component of MHC2 processing. Complement 3 is very highly upregulated. These are both co-localized to microglia themselves in the immediate area of the inclusion. And then we also see this, we can see with controls that a monomer injection does not cause the same inflammatory response. We can see this interesting sort of shift in crosstalk in that the initial response of C3 in microglia to the inclusions is happening at 2 and 4 months, but by 6 months, we still see a massive upregulation of C3, but by now it's the astrocytes that are expressing this gene and not the microglia. So we've become quite suspicious when reading the literature when people talk about C3 as astrocytic, especially when you're talking about aggregation, response to aggregates and pathology in neurodegenerative disease. So, when we first saw the C3 upregulation in our, GDPCR results, we assumed it was astrocytes like everyone else does. So we're big proponents of you need to really determine, because depending on the time and the relationship to the pathology, it could be microglia or astrocytes. Ultimately, we have sort of identified like a aggregate associated microglial signature of, in the immediate vicinity of inclusion, the microglia are regulating these genes. We've started to look, done some work looking at the whole transcriptome of inclusion-bearing microglia. And again, this is at 2 months. We spend a lot of time trying to figure out what's the most appropriate way to do a transcriptomic study. We're looking at a very localized region. So doing a cell-seq approach, we would have to batch many rats together to get enough tissue to do that experiment. We also know that single nuclei seq is not particularly appropriate for looking at microglial associated genes. So we ruled that out. And so we basically came up with, we're doing bulk seq, and we have done bulk seq, but we've tried to do it in the most refined way. So in this approach, we would use THGFP expressing rats. Using them allows us to visualize the contours of the nigra and do the best possible laser capture micro dissection that we can. And then again, to remind you, when we're capturing the substantia nigra in a PFF injected animal at 2 months, we're not only getting the PSIN, the enriched population of PSIN positive dopamine neurons, but we're also getting the immediately adjacent reactive microglia. And so doing that approach, we see that the upregulated transcripts are mainly associated with the immune response pathways. The downregulated transcripts were associated with neurotransmission and dopamine pathways. And one interesting thing that we identified by doing this approach in both male and female rats is that we see a sex difference in that female rats appear to not seed as efficiently as male rats do. So there's actually a slight reduction in that. And so that's cautionary for us in how we power our future studies looking at sex as a biological variable. Very recently, my graduate student, Jacob, who will be graduating in 2 months, we've tried to leverage this concept of we have early inclusions and later inclusions. This is all happening before degeneration is occurring. And we know that later inclusions are more Lewy body-like And we know that that is the time, right before the time when nigral neurons will begin to degenerate. So if we look at the transcript differences between what's seen at 1 month and what's seen at 2 months, can that help us identify sort of like linchpin genes or linchpin pathways that may be responsible for that conversion from I'm a neuron that's relatively healthy and I have an inclusion from a neuron that's beginning to degenerate and show cell death pathways. And so this is just showing volcano plots for so these rats are 1 month after PFF injection and 2 months after PFF injection. They both have the same amount of PSIN positive inclusions, but these at 2 months, we have relatively more mature inclusions. And we can see the gene expression profile at 1 month. There's more genes that are upregulated. A lot of genes upregulated with a low magnitude of upregulation, and more genes that are downregulated. And at 2 months, you can appreciate how we're starting to get fewer genes involved, but they're more spread out. And so when we look at genes that are uniquely upregulated at 2 months, that's where we think that could be a key inflammatory linchpin gene. And we're starting to look at some of these targets, localizing them to microglia. If we still continue to have supportive data, doing some sort of, addition, subtraction experiments to see whether we can actually show that these genes are important to, the neuroinflammatory response. So how can we use this model further, and how are we still using this model? We think about, if you believe that the prion-like hypothesis of pathological spread is driving Parkinson's disease, then you can use this model to study uptake mechanisms and perhaps how could you prevent uptake of subsequent transmission of pathological ASIN. And if you look at, do manipulations prior to PFF injections, you can look for therapies that could eventually perhaps prevent fibril uptake. If you look between months 0 and 2, where we have the peak in cortical inclusions, nigral inclusions, nigral MHC2 response, you can look at a lot of the neuroinflammatory questions that we've been looking at. What's the response of the brain and of the neurons themselves to the inclusions? And what's the response of the neighboring glia? And you can look at therapies that could slow or clear inclusion formation. And then if you look at later time points, that's when you can look at mechanisms that could prevent degeneration after inclusions form, which obviously would be very important for Parkinson's disease for patients that present with inclusion pathology already, as well as therapies to prevent subsequent degeneration. One way we've looked at this is to look at whether we can prevent fibril uptake and prevent inclusion formation and subsequent degeneration. We've worked basically with the premises of if this transmission of pathological ASIN is driving this dysfunction and degeneration, then therefore strategies to prevent its uptake would, be a good strategy to be neuroprotective. So we've looked at a particular target called TM9SF2, transmembrane 9 superfamily 2. This was identified by Mark Cookson's lab in a cell culture screen of receptors that if silenced, could you prevent uptake of preformed fibrils? And TM9SF2 rose to the top of targets that if silenced, they didn't get inclusion formation. We used an shRNA approach in rats to knock down TM9SF2 prior to doing the intracerebral PFF injection. And then we looked at TM9SF2. Then we looked to see confirming whether we had knockdown, and also more importantly, if we got rid of TM9SF2, could we prevent the accumulation of PSIN in the nigra at 2 months? And so here's just TM9SF2 is abundantly expressed in the nigra, and with our knockdown in red, you can see the loss of the red signal with our shRNA vector for TM9SF2. And when we look at 2 months, we can see significantly fewer inclusions. And so this is suggesting to us that, should this be truly an important mechanism? I'm couching myself because I'm not quite convinced. But should it be important to prevent second order spread of pathological ASIN, perhaps starting in TM92, which is enriched in the substantia nigra, could be a practical approach. We also have looked at as I mentioned, we've started to look at the role of corticostriatal synucleinopathy specifically on cognitive dysfunction. So again, this is ongoing work with my collaborator, Sheila Fleming. Here we're leveraging the fact that, again, we can have two conditions of equal nigrostriatal degeneration in the model, depending on the injection site. But if we inject in animals in the nigra, we'll have no cortical synuclein pathology, but in the striatum, we will have abundant cortical striatal pathology. And so we've started comparing the responses of these different surgical injection paradigms in a number of cognitive tests. And in particular, Sheila's doing a lot of work now with operant tasks, looking at cognitive flexibility. And so what she's seeing in rats that, have nigrostriatal synucleinopathy only, so resulting from the nigral injections, they are much less impaired compared to rats that have corticostriatal synucleinopathy. So we're trying to dissect what's the role of the loss of the nigrostriatal system. Is there a separate role for the corticostriatal inclusion pathology? And so we're definitely seeing that corticostriatal synucleinopathy is associated with this significant dysfunction in cognitive flexibility. And so, we're continuing on with larger groups and additional, but we're excited about the fact that we can sort of delineate the role of the synucleinopathy in striatum to these behavioral tests. And then I think this is the last data slide. The very last, most recent thing that we've been working on in collaboration with some of you here at University of Michigan is that we're developing a rat model of dementia with Lewy bodies. And so this is with my collaborator at Michigan State University, Scott Counts. Scott's worked for many years in this rat model of Alzheimer's disease, the TG34419 rat model. This is a model which develops plaques and in some, at advanced ages, neurofibrillary tangles, as well as degeneration in multiple regions. And what we're doing is we're basically combining this rat model with our PFF injections and looking to see whether if we induce co-pathologies, can we, what are the effects on each pathology and what are the effects on cognitive and motor behaviors. We'll be looking at various PET imaging markers here with our collaborators at U of M. And a number of behavioral markers with Sheila Fleming. And we're just starting this work. These studies take a while to go because you need to age the rats out for a period of time to induce these co-pathologies. But what we're seeing, so over here on the right is from a manuscript from Marsh and Blurt and Jones, and this is showing, co-pathologies of phosphorylated alpha-synuclein and A beta in a human Alzheimer's disease case. And this is essentially, you get these clusters of PSIN and A beta. And this is what we're seeing in the hippocampus of one of our Congo PFF AD rats. We're seeing this similar, we were struck by the similarities of how these profiles look. So we definitely we know that we can induce a model where there's co-pathologies, and now we're asking questions about what are the effects of these co-pathologies. In general, we have this injection strategy where we're timing the PFF injections to coincide with the pathology that is normally expressed in these AD rats.
Is dorsal striatal injection or?
Yes. We have 3 injection sites in the striatum. We've optimized in our original paradigm that was more focused on denervating the nigrostriatal system.
Right,
We didn't get a lot of inclusion pathology in the cingulate cortex.
Right.
And so now we have a 3rd injection site that's more rostral, that is the area that is most heavily innervated of the cingulate cortex. So tried to optimize, make sure that the pathology is supposed to be, is where it needs to be for those comparisons. We think that the cingulate cortex and the amygdala actually in these rats are going to be the two areas where there's the most abundant co-pathology, at least for Lewy bodies and for Pearson and plaques. So I'd just like to sum this up by some of what I see are the advantage and limitations of the rat alpha-synuclein preformed fibril model. Advantages is that the alpha-synuclein levels, are normal. It's not alpha-synuclein overexpression, and that's more analogous to sporadic PD. And I would also argue that even in cases of duplication and triplication, those are in Parkinson's disease, those are, those levels are estimated to be about 2 to 3-fold higher than sporadic Parkinson's disease. So again, alpha-synuclein overexpression in most models is not recapitulating the disease. The inclusions that are formed recapitulate multiple features of Lewy bodies, including the later stages where organelles and lipids are part of the Lewy body structure. The inclusions precede and predict the magnitude of degeneration. So we can tell if we do cohorts where we're looking at the same injection strategy, same quantity of initial fibrils, certain amount of inclusions at 2 months will predict a certain magnitude of degeneration at 6 months. We see widespread inclusion formation, not as widespread as some of the transgenic mice, but definitely depending on where you inject, you can definitely get multiple structures with inclusion pathology, or you can target it to a specific circuitry if you want to ask those questions. We see degeneration, neuroinflammation, motor and cognitive dysfunction. We like, although some could say this is a disadvantage, these relatively prolonged stages of pathology where there's this relatively discrete events going on, so you can ask different questions. And just as like a sidebar, I've worked with 6-hydroxydopamine, AAV vectors to overexpress alpha-synuclein. Once you get your initial material quality control, this has been the most consistent model that we've ever worked with, that I've ever worked with in 20 years of doing particle models. So, we don't have surgeries that don't work, and that would happen a lot with different batches of 6-hydroxydopamine. Certainly would happen a lot with AAV vectors, and so I like that consistency. Limitations is that you need to find a good source of preformed fibrils that are quality controlled. You need to sonicate them to the right size. If you're looking for a good source, I can hook you up with Dr. Joe Patterson, who is an excellent source of these quality fibrils. Some people don't like to do surgeries, so that could be a limitation. There is a lower magnitude of nigrostriatal degeneration and, by extent, neuroinflammation compared to some other models. But I guess I don't want to couch all these limitations because I'm trying to be transparent. But I guess I would say that perhaps that's a more disease analogous magnitude of neuroinflammation. The motor and cognitive deficits are subtle, so you do need larger group size, and that increases costs. The long time course makes things more expensive, especially if you're looking for degenerative endpoints. A lot of our experiments looking at inclusion, impact of inclusions, those are done over 2 months, so not too long. If you're looking for a model of second order spread of inclusion pathology, I do not believe this is the model. And then there's limitations that apply to all rat models, and that we all know rats are bigger, more expensive. There's fewer, transgenic tools that we can use to investigate hypotheses. So that's everybody. That's everybody in my laboratory, almost everybody, with a few new additions. And they're the team and the energy behind everything that I just showed you. Thank you.
So Carol, what's different about the models that claim they get second order spread?
Well, there's a few papers. Well, some of it I think is that we don't know all the connectivity of the brain.
Mm-hmm.
So it's not clear that truly if one structure doesn't innervate the other. But some papers will claim second order spread and they'll show you like one zoomed in image of a piece in like no context. And so to me, that's not convincing. I don't think as many papers claim that there's definitive second order spread as there are people that loosely talk about it and are maybe referring to templating as spread or, you can say you inject an striatum and you see inclusions in the nigra, but that's not spread, right? So a lot of people talk loosely about the spreading throughout the brain. I think initially this was supposed to be a model to substantiate Brock's prion hypothesis, and just, that's not empirically what people have said.
But there are groups that claim, I mean, like the GI injections that you get spread way up into that claims that's Brock-like, injecting the olfactory bulb.
Yeah, the GI pathology, their death pathology is done really well, but a lot of people don't see it, right? So it's pretty inconsistent. The GI stuff confuses. Sometimes the time course of sometimes after an injection into the duodenum, they're showing that nigral neurons are dying faster than if you inject it in the striatum, right? And there's just some things that I think the problem we have is that, alpha-synuclein is everywhere and it can be triggered by many things, including oxidative stress, including inflammation, including the neuron that's innervating me is perturbed. So I think definitely there's evidence of progressing along circuitry that PSIN pathology in a longitudinal way can be observed in this progression, but it still does not prove that a pathological form from the previous neuron jumped to the second neuron. So I guess that's what I'm advocating, that certainly in cell culture, in some conditions, you can definitively show that happened to some degree, but I think there are, too many other, if alpha-synuclein wasn't in the next neuron up the chain, you could say, it came from somewhere, but it's already there. So it's hard to prove, what caused the pathology in the first place.
You ever see T cells in your rats?
We haven't, we're trying to get the T cells to stain. It's a little difficult. We're having some trouble with that. We do see transcript for T cells. And certainly Ashley Harms's work, mostly in the aviation model, but she does have one study in the preformed verbal model. She's seen infiltration of T cells past a certain.
Carrie.
The injecting PFF in maybe like some of the infinite familial models to see if the PFF exacerbates kind of what happens if you start with a sick dopaminergic neuron, leading to the nigrostriatal degeneration rather than the degeneration occurring after the inclusion formation.
Yeah, there's a number of studies, mostly in mice. Mostly they've done look at like A53T, alpha-synuclein overexpressing mice. I think definitely there's an exacerbation. Sometimes in those models, it's hard to delineate about whether it is due to many of those models again are overexpression models. There's newer models that are knock-in models that we're working with right now. We have a rat A53T knock-in model looking at human PFFs, mouse PFFs, seeing whether there's any exacerbation of inclusion pathology or degeneration or any other type of phenotypes. But there's definitely some data out there, mostly again from the transgenics overexpressing, but even if you compare like wild type alpha-synuclein overexpressing mice with A53T or A30P overexpressing mice, and you introduce fibrils into that environment, there won't be more inclusion pathology when the, once the particular mutation.
What about, like non-synuclein familial?
Yeah. Aaron Moore looks at this in LARC2. He has a LARC2. I think he has mouse and rats. So yeah, people are trying to use this as a, can we use this as a tool to understand why these mutations are, promoting, fibrillization or promoting, synuclein pathology and degeneration.
But LARC2 doesn't always do that, however. So.
With in terms of like including and inducing pathology, yeah.
I mean, there's probably as many as 1/3 of LARC2 patients don't have secondary pathology.
Right, similar to Parkin, right? Parkin, there's
Parkin has almost zero.
Yeah, zero.
Zero.
Right?
I mean, Parkin is probably, I mean, again, this gets into the argument about what's PD, but Parkin is a mitochondrial disease, which is a distinct phenotype.
Right, but yeah, and there's synuclein pathology.
Right, almost.
There's a question in the chat. Hi Carol, nice presentation. Can I ask you what happens in terms of aggregation in rodents completed from either micro cells or T cells? Do they still form LB-like structures, or these cells require to form B-like structures?
So we tried to do that experiment. So we tried to do it. There's drugs to CSF R1 inhibitors, like pexidartinib, Plexicon drugs, to deplete microglia and see, so if microglia are involved in degeneration, let's take them out of the picture and see if degeneration is happening. So first, just to clarify, is that in cell culture, if you seed inclusions in neurons in cell culture, they will become dysfunctional and die. So they don't need the innate or adaptive immune system for this to be a toxic event in and of itself. So what we're trying to understand is that do inflammatory pathways, contribute or hasten that degeneration or include, induce spread. And so we did an experiment that's we published in rats where we used the CSF1R inhibitors to deplete microglia, but we ran up against the fact that in rats, those drugs don't work as well. So we only depleted like 50% of the microglia. And what we found is that we could deplete 50% of the homeostatic microglia, but the neighboring immediate reactive microglia, they were there. They didn't budge. Quantitatively, they stayed the same. And so we weren't able to change that population. What that is interesting to us is that's the same phenotype that's seen. That's a phenotype associated with efferocytosis, which is when microglia are eating a whole cell, eating an apoptotic cell. It's most extensively characterized during development in the retina, where you need a anti-inflammatory way to clear neurons that are overborn in development and need to be cleared. A process called efferocytosis. The microglia will just come and completely engulf the neuron that's apoptotic neuron, and those types of microglia undergoing efferocytosis don't depend on CSF1R receptor activation for survival any longer. So we didn't get to the experiment we wanted to do because we couldn't clear enough microglia, but the response to the drug gave us a clue of what else those microglia could be doing there. So that was kind of a convoluted answer. I hope that was kind of clear. But other people, have tried in mice to clear microglia, and are showing that it does impact the magnitude, not of the initial seeding and the inclusions, but impacts the rate of degeneration that's after the inclusion seeding. So that was a convoluted answer.
Did the degeneration change in your model, even though you've only completed
Yeah,
half?
so we saw no change in inclusion number, we saw no change in degeneration, but we never got rid of that most important population of microglia that were immediately adjacent. So we were unable to answer that question.
Okay. Micro injection versus the striatal injection, so you don't get PFC degeneration in pathology, but you get other regions that are projecting substantia nigra that now start to show some pathology.
Yeah, there are some brainstem nuclei that are showing pathology that you get, yeah, PPM for sure. Interestingly, like you can see, because there's a greater preponderance of innervation from the basolateral nucleus to the striatum, but more of this of the amygdala to the striatum than the central nucleus mostly innervates the substantia nigra. So you can see the pathology showing up at different regions of the amygdala. So
What about the striatum itself? I mean, I predict some striatal neurons would get.
we see, you can see PSIN pathology in the striatum after intranigral injection with that's in the terminals from the nigrostriatal system. So you see neuritic pathology in the striatum, but you don't see striatal pathology.
Expect to see some striatal neurons. Well, let's thank Carol for really excellent.